Hello everyone! My name is Naila MASTURA, but you can call me Teacher Nai. But wait, we are not alone in this exciting learning journey because I have my AI assistant named Jessie. Hello Jessie!
Hello Teacher Nai.
So, are you ready for another exciting learning adventure? Well! Today, we are going to embark on a fascinating journey through the microscopic world of multicellular organisms. We'll explore the intricate interaction between cells and their environment, tracing the evolutionary journey from the single-celled life to the complex organisms we see today.
Wow, that's exciting!
Are you ready, class?
Yeah!
Vamos, y aprendemos! So let's start at the beginning. The earliest life on Earth, dating back 3.5 billion years, consisted of simple single-celled organisms for over a billion years, and these single-celled life forms thrived, and some lineage, like eukaryotes, developing increasing molecular and cellular complexity. However, around 600 million years ago, the first multicellular animal known as metazoans emerged. This was a pivotal moment in the history of life, marking the transition from single-celled to multicellular existence.
What triggered multicellularity?
What drove this incredible shift to multicellularity? Scientists believe that environmental changes like an increase in atmospheric oxygen played a key role, and this oxygen increase led to a sudden burst of evolutionary diversity starting around 500 million years ago, and it also thought that radiation played a significant role as a larger multicellular organism could better defend themselves and capture prey. But to become a multicellular, organisms needed a way for cells to work together. They needed to recognize, adhere, and communicate with each other. This required a number of crucial innovations, which is the cell-to-cell interactions. Cell-to-cell interactions have three types, which are the cell recognition, cell adhesion, and cell communication.
Can you explain these three?
Of course, Jessie. So, cell recognition, where cells needed to be able to recognize and distinguish between other cells. On the other hand, cell adhesion, cells had to develop mechanisms to adhere to one another, forming cohesive tissues. And cell communication, where cells needed to communicate effectively to coordinate their activities and function as a unified organisms.
What is genome sequencing?
So when we say genome sequencing, it's like reading the complete set of instructions for building and maintaining an organism. It's like the process of determining the order of 4 chemical building blocks or bases that make up an organism DNA, which are the adenine, the guanine, the cytosine, and the thymine. Genome sequencing has helped us to understand how this happened, and genome sequencing has provided a valuable insight into the evolution of multicellularity and the studies of choanoflagellates. It is the single-celled organism considered to be close relatives of animals have been particularly illuminating.
How did the presence of molecules involved in cell adhesion, signaling, and extracellular matrix formation in Monozyga brevicollis contribute to the evolution of multicellularity?
So Jessie, the genetic blueprint of Monozyga brevicollis, a single-celled organism closely related to animals, reveals the existence of numerous molecules that play key roles in cell-to-cell interaction and the formation of the space between cell These molecules, which are also found in animals, were present long before the evolution of multicellular animals, and this suggests that these molecules originally used by single-celled organisms for tasks like sensing prey or attaching to the surface might have been repurposed to facilitate the development of multicellularity.
What do you think is genome duplication?
Genome duplication is an event likely played a crucial role in the diversification of molecules, leading the evolution of new functions in the newly evolving metazoan phyla. Now, let's shift our focus to the environment surrounding cells. While plasma membrane defines the boundary between a cell and its environment, the materials outside the membrane are crucial for cellular life. Most cells in multicellular organisms are organized into tissues where cells maintain a specific relationship with each other and with the extracellular material surrounding them.
Can you tell me about the cell-environment interactions?
Of course! These interactions are essential for various cellular processes, including the migration, the growth, and the differentiation. They also determine the three-dimensional organization of tissues and organs during embryonic development.
What about the extracellular matrix?
One of the most important components of the extracellular environment is the extracellular matrix, or ECM. This complex network of molecules provides structural supports, regulates the cell behavior, and facilitates the communication between cells.
Do you know glycocalyx?
Yes. When we say glycocalyx, it is the layer of carbohydrates projecting from the plasma membrane, and it is a part of ECM. It plays a role in cell recognition and cell adhesion. The ECM also contains various fibers such as collagen and elastin, and proteins such as laminin and fibronectin. And these molecules interact in specific ways to provide a structural support and regulate cell behavior.
What about cell surface receptors?
Cells interact with their environment through the specialized receptors on their surface, and these receptors bind to specific molecules in an ECM, transmitting the signals into the cell. The interaction of the cell surface receptors with the ligands initiate a cascade of events known as signal transduction, which ultimately alters the cellular behavior.
Wow, that's amazing!
Thank you, Jessie! So class, understanding the evolution of multicellularity and the intricate interaction between the cells and their environment is crucial for advancing our knowledge in the fields of medicine, biotechnology, and tissue engineering. By unveiling these complex processes, we can unlock new possibilities for treating diseases, developing innovative therapies, and engineering tissues and organs. Thank you for joining us on this journey through the microscopic world of multicellular organisms, and I hope you have learned in this lesson.
Hi everyone, Assalamu alaikum. I hope you're having a great day today. So by the way, I am Nafla Kalanduyan and I am the second reporter of this group. And at this video, I will be discussing to you the engineering linkage organoids and its interaction between cells and their environments. And of course, the interaction of cell with extracellular materials, and of course, its interaction between cell and their environments. So, let's start.
Engineering linkage organoids. So this is the idea of growing human organs in a laboratory, and it might sound like the stuff of a science fiction, but in fact, the engineering of organoids, three-dimensional cellular structure that resemble organs in both development and organization, has made significant advances in recent years and has a numerous promising applications in the lab as well as the clinics. So there are several different types of methods that support the growth of 3D cell cultures. One of them are the cells. So these cells are typically derived from stem cells, which can be tissue-specific stem cells from a mature organism such as stem cells from an adult intestinal crypts. So there are also embryonic stem cells or induced pluripotent stem cells. So these cells are then grown in a thick gel-like medium containing a complex mixture of extracellular matrix proteins and other components that encourage cellular growth. The most well-known is sold as matrigel. Depending on the organ that is being recapitulated, there are different cocktails of growth factors and additional extracellular matrix component must be added during the organoid development. So the end result is a small clump of tissue that can often approximately a full-size organ in different ways. So I'll show you later on the image of that full-size organ in a different way. So in recent years, researchers have made progress in developing organoids from a variety of different tissues including the intestine, stomach, pancreas, optic cup, and brain. How closely these organoids match with the parental tissue varies, but typically organoids can recapitulate many aspects of 3D such as architecture, tissue development, and of course cellular differentiation. So in the case of intestinal organoids, for example, villus-like epithelia with crypt-like domains and essential lumen are apparent. Brain organoids develop layers of neuronal cells that mimic the organization of the cortex. Having organs in a dish provides a multitude of opportunities for drug developers, clinicians, and scientists. So, organoids have been used to study diseases and test new drug therapies. So, for example, the brain organoids were used to study microcephaly caused by a Zika virus, and intestinal organoids naman create from a patient have been used to model and treat cystic fibrosis. So since yung organoids can be derived from the individual adults, it also holds promise for the development of personalized medicine. So it means in the near future, clinicians may be able to grow a series of organoid cultures as a body of chip that can be used for individualized and high throughput testing of potential therapeutics. So this is an image of a brain organoid through the fluorescent microscopy. So the neurons here are labeled green and are enriched in the outermost layer of the cortex, while the neural stem cells are labeled in pink. Okay, so now let's proceed to the interactions of cells with extracellular materials. So, as you learned in the section 7.1, which is discussed by my co-reporter, Ms. Naila Mastura, the components of the ECM such as fibronectin, laminin, proteoglycans, and collagen are capable of binding to receptors situated on the cell surface. So, as in figure 7.5, the most important family of receptors that attach cells to their extracellular microenvironment is the integrins. So we will know more about the integrins. Integrins is a family of membrane proteins found only in animals. So it plays a key role in integrating the extracellular and the intracellular environment. So on the outside of the plasma membrane, integrins bend to a remarkably diverse array of molecules that are present in the extracellular environment. So, on the intracellular side of the membrane, integrins interact either directly or indirectly with dozens of different proteins to influence the course of events within the cell. So, the integrins are composed of two membrane-spanning polypeptide chains, an A chain and an B chain. So, this is the two membrane-spanning polypeptide chains, an A chain and a B chain that are concovalently linked. So, according to my research, 18 different A subunits and 18 different B subunits have been identified. Although theoretically more than 100 possible pairings of A and B subunits could occur, only two dozen different integrins have been identified on the surface of cells, which is each with a specific distribution within the body. So this classification of integrin receptors based on recognition of RGD sequence was published in 2001 and display a highly unexpected feature. rather a standing upright as predicted, the integrin AB was bent dramatically at the knees so that the head faces the outer surface of the plasma membrane. So to understand the significance of this bent structure, we need to explore the properties of integrins. So this 2 is the integrin in the bent. So we have here the A, which is the schematic drawing of a complete integrin in the bent, which is inactive conformation and a corresponding electron micrograph of the extracellular portion of a similar molecule. And of course, we have also the B bent, which is a schematic drawing of an integrin in an active conformation and bound to a ligand, in this case talent. Binding of talent to the small cytoplasmic domain of the B subunit induces a separation of the two subunits and conversion of the active conformation. Activated integrins typically become clustered as the result of interaction of their cytoplasmic domains with the underlying cytoskeleton. So now let's move on to the role of integrins in platelet aggregation. So we have A and B. So the role of A is, it is the blood clots that form when platelets adhere to one another through fibrinogen bridges that bind to the platelet integrins, while the B is, this is the process where the presence of synthetic RGD peptides can exhibit blood clot formation by completing with fibrinogen molecules for the RGD building sites of platelets. So now we are here in the steps in the process of cell spreading. So scanning electron micrographs showing the morphology of mouse fibroblasts at successive times during attachment and spreading on glass cover slip cells were fixed after A 30 minutes, B 60 minutes, and C 2 hours, and D 24 hours of attachment. So now we're here at the focal adhesion. So these focal adhesions are sites where cells adhere to their substratum and transmit signals in both directions across the plasma membrane. So we have the picture A, picture B, and picture C. So in A, this picture is cultured cell that has been stained with fluorescent and antibodies to reveal the location of actin filaments, which is color green, and integrins, which is color red. So the integrins are localized in small patches that correspond to the sites of local adhesions. So in the second picture, which is the B, the cytoplasmic surface of a focal adhesion of a cultured amphibian cell is shown here after the inner surface of the membrane was processed for quick freeze deep etch analysis. Bundles of an actin filaments are seen to associate with the inner surface of the membrane in the region of a focal adhesion. While the third picture, or in the third picture, this is a schematic drawing of a focal adhesion which shows or showing the interactions of integrin molecules with other proteins on both sides of the lipid bilayers. So the binding of extracellular ligands such as a collagen and fibronectin is thought to induce informational changes in the cytoplasmic domains of the integrins that cause the integrins to become linked to an actin filaments of the cytoskeleton. So there are linkages with the cytoskeleton's lead. So in turn to the clustering of integrins at the cell surface, linkage with the cytoskeleton are mediated by various actin binding proteins such as tallin, which shown in the picture color yellow, and an A-actinin, which is shown as a color purple, and that bind to the B subunit of the integrin. So during the process of local adhesion formation, tallin undergoes a conformational change that exposes binding sites on tallin's rod domain. So the binding of vinculin, which is that light green, to these exposed sites promotes the assembly of additional actin filaments. So the cytoplasmic domains of integrins are also associated with protein kinesis such as FAK, which is focal adhesion kinase, and also it is shown as color orange, and of course SRC, the attachment of the integrin to the external or extracellular ligand, can activate this protein kinesis and start a chain reaction that transmits signals throughout the cell. So the association of myosin molecules with the actin filaments can generate traction forces that are transmitted to sites of cell substrate attachment. So now we are here in the experimental demonstration of forces exerted by focal adhesion. So as you can see here in the picture, that picture is a scanning electron micrograph of a smooth muscle cell sitting atop a bed of flexible, elastomeric micropores with tips that had been coated with fibronectin. So the attached cell is seen defecting the position of muscle pose. So the degree of movement of a particular pose reflects the magnitude of the traction forces exerted by the cell on that pose. Okay, so we have here the hemidesmosomes. So it is a focal adhesion are most commonly seen in cells grown in vitro, although similar types of adhesive contacts are found in certain tissues such as muscle and tendon. So within the body, the tightest attachment between a cell and its extracellular matrix is seen at the basal surface of epithelial cells. So where the cells are anchored to the underlying basement membrane by the specialized adhesive structure called the hemidesmosome. So, hemidesmosome contain a dense plaque on the inner surface of the plasma membrane with filaments coursing outward into the cytoplasm. Okay, so now we're here at the measuring the force necessary to activate integrins. So in this experiment, an integrin's ligand was attached to a glass substrate using a tether that would rupture once a specific force is applied. So the mammalian cells were allowed to spread on the glass, and the cells were stained for actin, which is the actin is color green, and a focal adhesion marker, which is vinculin, and the color red. And of course, when tethers of the rupture strength of 43 piconewtons were used, the integrins were not activated and the focal adhesion did not form. However, the tethers of a rupture strength of 56 piconewtons resulted in integrin activation as seen through the formation of focal adhesions. Okay, so now we are here at the last part of my topic, which are the hemidesmosomes. So, hemidesmosomes are differentiated site at the basal surfaces of epithelial cells where the cells are attached to the underlying basement membranes. So as you can see, we have the picture A and the picture B. So the picture A is electron micrograph of several hemidesmosomes, which is it's showing the dense plaque on the inner surface of the plasma membrane and the intermediate filaments projecting into the cytoplasms, while the B pictures is a schematic diagram which is showing the major components of a hemidesmosomes connecting the epidermis to the underlying dermis. So the A and B integrin molecules of the epidermal cells are linked to cytoplasmic intermediate filaments by a protein called plectin. So that plectin is present in the dark staining plaque and to the basement membrane by anchoring filaments of a particular type of laminin. So a second transmembrane protein is also present in hemidesmosome. So the collagen fiber are also part of the underlying dermis. So that's the end of my discussion. Thank you so much.
Hello everyone, I am Saadra DIMALNA, and this time I'm going to discuss to you about the interaction of cells with other cells. So let's start. Early research involved dissociating cells from developing organs and observing their reaggregation and culture. Cells from different organs initially form mixed clumps, but eventually sort themselves out, adhering only to cells of the same type. This sorting process is thought to be driven by specific interactions between cell surface molecules. Four distinct families of integral membrane proteins play a major role in mediating cell-to-cell adhesion. Number 1, selectins. Number 2, certain members of the immunoglobulin superfamily. Number 3, certain members of the integrin family. And number 4, cadherins. Next, we have selectins. Selectins are like sticky notes for cells, specifically for leukocytes or the white blood cells. They recognize and bind to specific sugar arrangements on the surface of other cells, like the lining of the blood vessels. So the role and inflammation. During inflammation, selectins help to slow down leukocytes as they flow through the bloodstream, allowing them to roll along the vessel wall and eventually migrate into the inflamed tissue to fight infection. There are three types of selectin: E-selectin that can be found on endothelial cells, P-selectin that can be found on platelets in endothelial cells, and L-selectin that can be found on leukocytes. So we have here an example of the role of selectin. So during a bacterial infection, selectins on the surface of blood vessel walls or endothelial cells bind to specific sugars on the surface of neutrophils, a type of white blood cell. This interaction slows down the neutrophils, allowing them to roll along the vessel wall and eventually migrate into the infected tissue to fight the bacteria. So next, we have the immunoglobulin superfamily or IGSF. So this family of proteins is primarily involved in the immune system, but some members also play a crucial role in cell-to-cell adhesion. The presence of Ig-like domains and cell adhesion receptors in invertebrates, or the animals without a classic immune system, suggests that these proteins originally evolved for adhesion and only later became involved in immune responses. Example: VCEM or vascular cell adhesion molecule, NCAM or neural cell adhesion molecule, and L1, also called L1CEM, are IGSF members that mediate adhesion between non-immune cells, particularly important in nerve development. Example of role of immunoglobulin superfamily. NCAM or neural cell adhesion molecule, a member of the IGSF, plays a crucial role in the development of the nervous system. It helps neurons to adhere to each other and to guide the growth of axons or the long projections of neurons to their correct targets. Next, we have cadherins. Cadherins are the glue that hold cells together, particularly cells of the same type. They form strong calcium-dependent bonds between neighboring cells. Role and development. Cadherins are essential for the formation of tissues and organs during embryonic development. They help to organize cells into cohesive structures and maintain their integrity. So types of cadherin: E-cadherin or epithelial, N-cadherin or neural, and P-cadherin or placental are some of the best studied cadherins. Example of role of cadherins: E-cadherin, a type of cadherin found in epithelial cells, helps to hold these cells together in a cohesive sheet, forming a barrier that protects the underlying tissues. In the development of the neural tube, which will become the brain and spinal cord, cells switch from expressing E-cadherin to expressing N-cadherin, allowing them to detach from the surrounding epithelial layer and from the neural tube. Next, we have adherence junctions and desmosomes. Adherent junctions. These junctions form belts around cells, connecting them to their neighbors near their apical surface. So the function is they link the external environment to the actin cytoskeleton, a network of protein fibers within cells, providing structural support and pathway for signals to travel from the cell exterior to the interior. Desmosomes. Desmosomes are disc-shaped junctions found in tissues that experience a lot of mechanical stress like skin and heart muscles. So the function is they contain specialized coherence and connect to intermediate filaments, another type of protein fiber within cells, providing strong structural integrity to cell sheets and preventing them from tearing apart. Example: adherence junctions are found in the lining of the intestines, helping to hold the epithelial cells together and forming a barrier that prevents the passage of harmful substance from the gut into the bloodstream. Desmosomes are found in the skin, providing strong connections between the cells that make up the epidermis. This helps to maintain the structural integrity of the skin and allows it to withstand mechanical stress. So that's all. I hope you learned something. Thank you.
Hi everyone, I'm IBAD Tanya A, and you can call me Mom IBAD. And today we're going to talk about the tight junction. Oops, not only tight junction, but also gap junction and plasmodesmata. Have you ever wondered how our bodies prevent harmful substances from entering our bloodstream? Well, tight junction play a key role. They're like a tiny seals between cells and prevent a leakage. So now let's define what is tight junction. So tight junction, also known as Zenole occludens, are specialized cell junction that form a continuous seal between epithelial cells and preventing the leakage of fluids and molecules between cells. They are crucial for maintaining the integrity of epithelial cells or epithelial tissue and regulating the passage of substances across these barriers. Just imagine your body is like a city made up of a lot of buildings, such as cells. For example, union cell. So tight junction are special seals between these buildings, preventing leaks and keeping things organized. They're vital for keeping harmful substances out and essential substances in. So they're found in many places like our intestine, where they stop bacteria from entering our bloodstream, and also in our brains from forming the blood-brain barrier. So okay, now let's talk about its structure. So as you can see in the image is a diagram illustrating the structure of the tight junction between two cells, such as cell A and cell B. So the cell A and the cell B, these represent two adjacent cells. Tight junction are the structure that connect these cells. So as you can see, nandiyan ang ating tight junction. So that's the barrier that prevent the passage of substances between the cells. And also there's a plasma membrane. So this is the outer boundary of each cell. The tight junction is formed by proteins embedded within the plasma membrane of the both cells. And also, there is a groups of tight junction proteins. So these are the cluster of transmembrane proteins, shown as the light blue ovals. So that are the main components of the tight junction. So these proteins is primarily claudins and occludins, which is interacting with each other across the extracellular space to form a tight seal. So occludins, this protein was the first integral membrane protein identified in tight junction. So occludins are tetraspan transmembrane proteins, meaning they have four transmembrane domains that span the cell membrane. So always keep it in mind that occludins are not only present in epithelial and endothelial cells, but also expressed in cells without tight junction, such as neurons, astrocytes, and etc. So this suggests that the occludins may have additional function beyond tight junction formation. So on the other hand, claudins discovered after occludin. Claudins are a family of transmembrane proteins that form the backbone of tight junction. They are responsible for the barrier function and the selective permeability of the junction. So the primary function of claudins is to form the core of the tight junction barrier, creating a selective permeability barrier that controls the flow of molecules in the intercellular space. So occludins and claudins are integral membrane proteins that play a crucial role in the formation and function of tight junction, which are essential structure in epithelial and endothelial cells. And also there's an extracellular space. This is the space between the two cells, as you can see in the picture. So the tight junction proteins create a seal within this space and preventing leakage. So as you can see in the diagram, it shows how tight junction proteins in the plasma membranes of the adjacent cells interact to form a continuous seal, preventing the passage of molecules and ions between those cells. So this seal is crucial for maintaining the integrity of tissues and organs and regulating the movement of substances across the epithelial barriers. Okay, next we're going to talk about its function. So tight junction are essential structures in epithelial and endothelial cells, acting as a barrier that control the passage of molecules between cells. So their main function include barrier function. So tight junction prevent the leakage of fluids and molecules between cells, creating a barrier that regulates the paracellular transport of substances. So this is crucial for maintaining the integrity of epithelial tissues and controlling the passage of nutrients. So this is crucial for maintaining the integrity of epithelial tissues and controlling the passage of nutrients, electrocytes, and other substances between the different compartments of the body. So in cell polarity, tight junction help establish a top and a bottom for cells, creating a polarity. So this is important for cells that need to have a different function on a different sites. For example, in the intestine, the top of the cell absorbs nutrients, while the bottom side faces the bloodstream. So last but not the least, maintaining tissue integrity. So tight junction help keep tissue strong and intact. They act like glue, holding cells together, preventing them from separating. So this is important for maintaining the structure and function of the organs. To sum up everything, tight junctions are essential for maintaining the integrity and function of epithelial tissues and regulating the passage of substances across these barriers. They are involved in a wide range of physiological processes, and their dysfunction can contribute to a variety of pathological conditions. So the tight junctions are tiny but mighty gatekeepers, ensuring our body function properly. Okay, so now let's move on to our next discussion, and that's the gap junction and plasmodesmata. So we're going to talk about its function, structure, and importance. So first, we're going to talk about the gap junction. So gap junction are specialized intercellular junction that directly connect to the cytoplasm of two adjacent cells. They are formed by the close opposition of the plasma membranes of these cells, with a narrow gap of about 2 to 4 nanometers between them. So this gap is spanned by connexin, which are transmembrane protein channels that allow for the passage of small molecules and ions between the cells. So now we're going to talk about its function. So the function of gap junction include direct intercellular communication, metabolic coupling, electrical synthesis, development and differentiation, and also tissue homeostasis. So in direct intercellular communication, gap junction allow for the rapid and direct exchange of ions, small molecules, and signaling molecules between cells. These allow for a coordinated cellular activity and rapid communication within tissues. Metabolic coupling, gap junction can facilitate the transfer of nutrients and metabolites between cells, ensuring a balanced metabolic state within a tissue. And then electrical synthesis. In some tissue, gap junction form a electrical synthesis, allowing for the rapid transmission of electrical signals between neurons. So this is important for a coordinate activity in our nervous system. And also in development and differentiate. So gap junction play a role in cell-cell interaction during the development and differentiation, influencing cell fate and tissue organization. So next is the tissue homeostasis. Tissue homeostasis, gap junction contribute to tissue cellular responses to environmental changes and maintaining the tissue integrity. So next, let's talk about its structure. So as you can see in the picture, there's a connexin monomer or connexin subunits, and hydrophilic channels or gap junction channels, and also intercellular space and plasma membrane. 1, 2, 3, go. So the connexin, these are the fundamental building blocks of gap junction. So each connexin is a hexameric protein complex composed of 6 identical connexin subunits. So connexin from 2 adjacent cells align to form a continuous channel, allowing for the passage of molecules between the cells. So the space between the plasma membrane of adjacent cells is called the gap junction channel or the hydrophilic channel. So this channel is approximately 2 to 4 nanometer wide and is spanned by the aligned connexin from the 2 cells. So connexin manometer is a direct cell-to-cell communication by allowing the passage of small molecules, including ions, metabolites, and signaling molecules, So they are essential for coordinating cell activity, development, and metabolic coupling. So that's the connection manometer or connection subunit. So now let's talk about its importance. So gap junctions are essential for a wide range of physiological processes, including nervous system. Electrical synapses formed by a gap junction ensure the rapid communication between neurons and crucial for coordinated neutral activity. And also importance in our heart. Gap junction facilitate the spread of electrical signals throughout the heart, ensuring the synchronized contraction, and also in smooth muscle. So gap junction allow for coordinate contraction in smooth muscle tissue, such as gastrointestinal tract. And also in development, gap junction play a critical role in cell-to-cell communication during the embryonic development and guiding tissue formation and organogenesis. So, understanding the structure and function of gap junction is crucial for understanding how cells communicate and coordinate their activities within tissue and organs. Now, let's discuss the plasmodesmata. So, what is plasmodesmata? So, plasmodesma in singular are microscopic channels that connect the cytoplasm of adjacent plant cells, allowing for the direct communication and exchange of molecules between them. So these channels are essential for plant development, growth, and response to environmental stimuli. So now let's talk about its structure. So as you can see in the image, there's two adjacent plant cells connected by a plasmodesma. So what are you waiting for? So let's break down the level components. So as you can see, there's a cell wall. So that's the outer layer of each plant cell, providing structural support. So the arrows indicate that substances can move through the cell wall, but not directly between the cell cytoplasm. And also there is a cytoplasm, the gel-like substance filling the cell, containing organelles and carrying out the cellular processes. So the orange line illustrates the cytoplasmic continuity through the plasmodesma. And also, there is a vacuole, a large fluid-filled sac within the cell involved in storage, waste disposal, and maintaining turgor pressure. And also, there is a plasmodesma. So, the central focus of the image is the plasmodesma. So, this is a channel that directly connects the cytoplasm of two adjacent plant cells and enabling communication and transport between them. So the diagram shows how the cytoplasm flow continuously through this channel. So the image highlights the plasmodesma's role in facilitating communication and transport between plant cells. Despite the presence of the cell wall that would otherwise embed direct cytoplasmic exchange. So the arrows show the direction of movement of substances through the plasmodesma. So now, how about we talk about its function and importance? So first, let's talk about its function. So these include the cell-to-cell communication. So plasmodesmata facilitate the direct exchange of molecules between cells, allowing for a rapid and efficient communication within plant tissues. This communication is crucial for coordinating growth, development, and responses to environmental cues, and also transport of molecules. So plasmodesmata allow for the transport of various molecules, including small molecules such as sugar, amino acid, ions, and other small molecules can readily pass through plasmodesmata. And also in proteins. So some proteins can pass through plasmodesmata, although their size and structure can limit their movement. And also in our RNA, our messenger RNA can move through plasmodesmata, enabling gene expression to be coordinated between cells and potentially influencing cell fate. And also in viruses. Some plant viruses can exploit plasmodesmata to spread from cell to cell. And also development of differentiation. So plasmodesmata play a crucial role in plant development and differentiation. So the formation and regulation of plasmodesmata contribute to establishment of cell-to-cell communication. Cell-to-cell communication networks that are essential for coordinating tissue growth and organogenesis. So last but not the least, its function in stress responses. So plasmodesmata are involved in plant responses to various stresses, including drought, salinity, and pathogen attack. So they facilitate the transport of signaling molecules and defense compounds between cells, enabling the plant to mount an effective response to these challenges. So now let's talk about its importance. So plasmodesmata are essential for plant life, playing a critical role in growth and development, stress responses, and nutrient transport. So in growth and development, they facilitate communication and coordination between cells, enabling proper tissue development and organogenesis. So in stress responses, they allow for a rapid communication and transport of defense compounds, enabling plants to respond effectively to various stresses, as we mentioned before. And also in nutrient transport, they facilitate the movement of nutrients and metabolites between cells, ensuring the proper nutrient distribution within the plants. So when we say intercellular communication, it is the process by which cells interact with each other and exchanging information and coordinating their activities.
Assalamu alaikum. My name is Alaiza. I'm LADSAK and I am the last reporter from the group 3. And my topic is cell walls and green cells, cell walls and plant terrestrializations. So our learning objectives: to know what is cell walls and plant terrestrialization, identify the cell walls, and explain what is cell walls. So first, what is cell wall? A cell wall is a structural layer surrounding some types of cells. Situated outside the cell membrane, it provides support, protection, and shape to the cell. So, in plants, ang cell wall is made of cellulose, which is type of carbohydrate. So this make plant cells rigid and strong. So because of the cell wall, plants like trees can stand tall and resist damage from external forces. So we have the function of the cell like what I read kanina. First one is support. So it gives the cell a strong framework para hindi ito mag-collapse. And the second one is protection. So it acts a barrier against harmful substances or physical damages. And the third one is shape. So it helps maintain the proper shape and cell kahit na ma-expose sa stress like water pressure. So makikita ang cell wall sa plant cells, fungi, bacteria, and some protists. So pero hindi ito makikita sa animal cell. So, in short, it's a tough layer na nagpapalakas sa cell structure. The next is what is plant cell wall. So, plant cell walls, the first cellular structures visible with a light microscope, have multiple vital functions. They provide structural support for individual cells and collectively act as a skeleton for the entire plant. So, cell walls also protect against damage and mediate cell-cell interaction. So, the plant cell wall is outer layer ng plants or plant cells na madaling makita gamit ang light microscope. So, it serves many important functions. First one is structural support. So, nagbibigay ito ng matibay na support sa bawat cell at nagsisilbing parang skeleton like what I've read a while ago ng buong plant. So, dahil dito, ang plant can stand tall and maintain their shape kahit subjected sa iba't ibang pressure tulad ng gravity or strong winds. So, next is protection. So, pinoprotektahan ng mga cell wall ang cells laban sa damage tulad ng physical impacts or environmental stress. So, it also helps regulate interaction sa pagitan ng neighboring cells, which is crucial for plant growth and communication. So, their structure is similar to reinforced concrete of fiberglass. So, with cellulose microfibrils providing the fibrous component embedded in a gel-like matrix composed of hemicelluloses, pectins, and proteins. So, ang structure ng plant cell wall ay parang reinforced concrete or fiberglass. So, ang cellulose microfibrils ay nagsisilbing fibrous material na nagbibigay ng strength habang ang gel-like matrix ay binubuo ng hemicelluloses, pectins, and proteins. So, ang combination ng hemicelluloses, pectins, and proteins is dahilan kung bakit ang cell wall ay matibay pero flexible pa rin. So, ang plant cell wall ang dahilan kung bakit ang mga plant ay matatag at kayang mag-adapt ng challenging environments. Hemicelluloses are branched polysaccharides that cross-link cellulose microfibrils. So, itong hemicelluloses is parang sila ang connectors na nag-hold sa cellulose fibers, which strengthens the cell wall. So, this cross-linking is essential para maging matibay pero flexible ang structure ng cell wall. Pectins are negatively charged polysaccharides that hold water, forming a hydrated gel. So, ang pectins is isa siyang negative charge polysaccharides na kayang mag-hold ng water kaya sila ay nagpo-form ng isang hydrated gel. So, this gel-like property na nagbibigay ng elasticity sa cell wall. So, making it the cell wall adaptable sa pressure changes. So, ito ang dahilan kung bakit ang cells ay hindi basta-basta natutuyo. So, the third one is proteins. Proteins mediate dynamic activities with expansions facilitating cell growth. So, ang proteins ay mahalaga para sa dynamic activities ng cell wall. Halimbawa, ang expansions ay isang uri ng protein na tumutulong sa cell growth by loosening parts of the cell wall. So, ito ay importante lalo na sa mga stages ng plant growth, kung saan kailangan mag-expand ng cells habang nananatiling structurally stable. So, in other words, ang combination ng hemicelluloses, pectins, at proteins ay nagbibigay ng strength, flexibility, at functionality sa plant cell wall. So, let's look at the picture. So, this is the plant cell wall. So, as you can see at the image A, meron siya nagkalagay na primary cell wall and middle lamella. So, ang primary cell wall dyan is yan yung outer layer ng plant cell na flexible, kaya nakakatulong ito sa growth ng cell habang bata pa yung plant. And the middle lamella naman is the layer between two plant cells. Ang middle lamella ay gawa sa pectin at nagsisilbing cement na nagdidikit sa neighboring cells that keeping the plant tissues intact. And the yung nasa middle picture naman, yung letter B is this is a close-up of the network of fibers sa loob ng cell wall. So makikita dito ang tightly packed cellulose microfibrils na nagbibigay ng strength at stability sa cell wall. So these fibers are arranged randomly in young cells but become more organized as the cell matures. So it ensuring strength and flexibility. So the third picture naman is the diagram ng molecular structure ng cell wall showing its key components. So meron diyang cellulose microfibrils. So these are the main fibers na nagbibigay ng rigidity sa cell wall, and hemicellulose naman is like what I've said is parang cross-linkers na kumokonekta sa cellulose microfibrils, making the structure strong but flexible. So, meron din dyang pectins. Pectin is responsible for holding water, kaya soft and gel-like ang consistency ng cell wall in some parts. And meron din dyang glycoproteins. So, ang glycoproteins, it helps in dynamic processes like growth and repair, supporting the flexibility and adaptability ng cell wall. So, plant cell wall is a complex structure na binubuo ng iba't ibang layers and components working together. So, it provides strength, protection, and support habang flexible enough to allow growth and interaction between cells. So, itong picture naman is the synthesis of plant cell wall micromolecules. So, as you can see at the first picture, the letter A, this is an electron microscope view showing the arrangement of cellulose microfibrils. So, the cellulose microfibrils are the main structural components ng cell wall. Makikita sa image na tightly packed ang fibers which gives the wall its strength and rigidity. So, the cross-linking ng mga fibers ay tumutulong sa flexibility ng cell wall habang nananatili itong matibay. And the second one na picture sa middle is this is a diagram illustrate the process of cellulose synthesis. So, the cellulose synthesis or cellulose synthase rosette, yung green oval dyan na structure ay matatagpuan sa plasma membrane. So, ito ang enzyme complex na responsible sa paggawa ng cellulose. So, microfibrils are produced as the cellulose synthase moves along the microtubules, yung blue lines dyan. So, it ensuring the proper arrangement ng cellulose fibers. So, ang coordinate action ng cellulose synthase at microtubules ang nagbibigay ng order sa formation ng cell wall components. So, yung third picture naman is this shows the Golgi apparatus of its role in synthesizing other micromolecules like pectins and hemicellulose. So, makikita dito na ang Golgi apparatus, yung black dots dyan, ay gumagawa ng mga polysaccharides na ginagamit sa cell wall. So, these molecules are transported via vesicle papunta sa cell membrane where they are assembled into the cell wall matrix. Green cells, cell walls, and plant terrestrialization. Algae initially live in oceans and fresh water, but around 450 million years ago, a population transitioned to land. This transition required several adaptations, particularly in cell wall structure. Compared to the other algae, CCG exhibit a rosette arrangement of cellulose synthase complexes and a higher density of cellulose in their cell walls. So these features, along with other cell wall innovations like pectins, are hypothesized to have been crucial for survival on land, providing resistance to desiccation and UV damage ultimately paving the way for the explosion of plant life on Earth. So the section concludes with a review question asking to describe two evolutionary innovations that facilitated this transition from water to land. So, ang algae ay karaniwang naninirahan sa mga tubig and sa dagat at freshwater noong una. So, ang paglipat sa lupa ay nangangailangan ng maraming pagbago sa kanilang cell wall structure. So, cell walls help protect the plant cells mula sa harsh condition ng lupa tulad ng desiccation or pagkatuyo ng lupa at UV radiation. So, this adaptation paved the way para makasurvive ang mga plant sa lupa and eventually to evolve into various plant forms. The CGA and cellulose synthase compared to other algae, ang CGA or Charophysian green algae ay nagdevelop ng unique features sa kanilang cell walls. The first one is the rosette arrangement of cellulose synthase complexes. So, ang mga enzymes na ito is responsible sa paggawa ng cellulose microfibrils which give the cell wall its strength and flexibility. Sa CGA, mas organized ang arrangement na ito which contribute to their ability to withstand land conditions. And the second one is higher density of cellulose. So, dahil mas maraming cellulose, mas matibay ang kanilang cell walls, kaya kaya nilang mag-survive sa lupa kahit under physical stress. So, bukod sa cellulose, ang pectin sa kanilang cell walls ay nagbibigay ng hydration and elasticity. So, these features were crucial para hindi sila mabilis matuyo at ma-maintain ang flexibility habang nag-a-adapt sila ng bagong environment. So, this combination of innovations ultimately paved the way for the explosion of plant life on Earth, leading to the diversity of plants we see today. So, ang transition ng algae mula sa tubig papunta sa lupa ay nangyayari dahil sa kanilang evolutionary innovations sa cell wall structure tulad ng rosette arrangement of cellulose synthase, pectins, and cellulose modification for protection and support, and these adaptations allowed plants to thrive and expand on land, transforming the Earth ecosystem.